Okay, so thank you for this invitation and the kind introduction. We're really excited to present the HGRepo Informatics framework, which we developed for the NIAID Division of Intramural Research for the COVID-19 Immune Response Consortium. So this is going to be a tag team. Team effort. And I'm going to start off with a description of the immune response research, really, because this is going to sort of inform you of how it actually affected the way how it influenced the way we deployed LabKey for HGRepo, and also the architecture, how did it influence it. So I'd like to also mention that we'll give you a brief view of the vision and summarize the presentation. Okay, so the background. The data for the COVID-19 consortium studies comes not from clinical trials, but it's really from natural history studies. And so what that means is the data is unstructured, right? Because they're coming from multiple hospital sites, both domestic and international. We have up to 14 now and growing. The data are all de-identified, however, with personal health information. There are several goals of the researchers here. Host genetics, and here we have whole genome sequencing data. We have the host response, and here you have gene expression, single cell multi-omics data, chromatin assembly, humoral signature, serology, and importantly, microbial genetics to understand what is the role of the microbiome in disease susceptibility, and how are the different strains influencing the severity. However, all of this data that's being generated has to definitely be linked back to the clinical data, the demographics, clinical profile, phenotypes, and tracking and linking and making sure that these are all then accessible to the collaborative researchers for integrative analysis. This is really key for any system that needs to be developed. So quick overview of the workflow. The consortium PIs have a memorandum of understandings with the medical institutions with whom they're partnering, receiving the samples and clinical data. They're received by over 20 PIs. So you can imagine the coordination that has to occur to distribute the samples, the clinical data to the research labs that actually perform the assays. And then they report back results to the consortium. So really the linking of all of this is so crucial and has to happen from the get-go. So these were some of what the requirements that guided us into defining and developing our scientific data management system. So there were several challenges, but also really opportunities that were part of this effort. So first of all, the researchers from the get-go designed a questionnaire, a COVID-19 patient screen. They have this sort of very well-formatted Excel document with definite sections for demographics, laboratory findings, etc., so that they would get the data, standard data in a standard format. That was their goal. However, this is a pandemic, so really not a clinical trial, right? The clinicians are more interested in treatment than they are in getting you all of the different data in exactly the format you want. And moreover, some sites have very good EMR capabilities, and others really have paper records. So really what we ended up getting is a combination of Excel formats that look nothing like the original, or even screenshots. So this is really a screenshot that had to be parsed to provide the structural data that was necessary for actually digitizing. And the data corrections came, supplemental data, and then what I want to emphasize here, we all have heard of the urgency, the COVID speed access that's necessary for the researchers to actually perform collaborative analyses. So we were sort of, really kind of like a wit's end. However, what we realized was there was really enormous opportunity here because everybody was really interested in working together. And who do I mean by everybody? Sample and clinical data curators, medical experts, data scientists and statisticians, data transformers, assay data experts, system development. Folks. So we all worked, and for sometimes we would work almost all day. We would be in contact with somebody or the other. So the system that was needed to really support this ecosystem, what we call, is something that would allow multiple data capture modes, because we wanted a sustainable system, script-based or user upload tables, or integrate with other existing sources, link patient samples to assay longitudinal data, access to diverse data sets, standardization, querying, sharing, reporting, visualizing charts and tables, the data, democratize data access for spreadsheets or APIs for programmatic access and scripting. And this is really important for enabling novel data science and distributed analyses. So really, the next generation system is what we wanted: collaborative access with role-based access, standard method of authentication, ability to publish synthetic and de-identified data sets so that we could then share the data broadly. So these were all key for us, and we looked around and we heard of LabKey, and we chose that for our clinical and sample data. We could store data in a structured, standardized, and analysis-ready data sets could be exported. But then for the assay data, since we really didn't want parsed data, but we wanted raw assay data files, we chose another data sharing platform that was outside of the NIAID firewall, easily accessible to users, and also that allowed file level sharing. But the idea is that the summary level data, assay data, is going to be brought back to LabKey for further browsing the data along with the clinical data. So I don't want to belabor this a lot here with regard to the timeline, but when we first heard of LabKey, it was in May, and we ended up deploying the enterprise version by the end of June. And then from July to now, we have this rich set of features. And really what I want to thank the LabKey, both from the acquisition personnel as well as from definitely the technical people who allowed us to implement this really creative solution that allowed us to support our COVID-19 ecosystem. So with that, I'm going to then pass off the baton to Jason, who's going to really demo, and hopefully you will see how all of this is captured in the system. So Jason?
Thank you, Sandhya. So just to recap, we had all of these different information sources and all of these different formats that were coming to us, as well as sample information. We had to transform all of that into kind of a standard, structured place. And so LabKey provided that foundation onto which we're putting all of this data. And so we're just listing here on this slide some of the things I'm going to cover in the next few minutes. And those include the fact that LabKey allowed us to define our data sets and sample properties. They allowed us to use certain common data elements like controlled vocabularies and data groupings, which I'll show you. And also we're able to build reports and views to our customer and the working group specifications. And there are cases where there's a lot of hand curating that's going on, but there are also cases where we said, if you have these two data points, you can do a calculation and infer this other data. So we have automated updates that are happening to some of our timeline data, and also data coming from the collaborative data sharing portal that Sandhya mentioned, where we have some of our assay data. So just marketing the ability to say there's also data available in this system is possible in LabKey. And lastly, NIH authentication was a big requirement for us, and LabKey really helped us sort out what we needed to do to get that set up. And all of this is done with folder and role permission. So I'm going to share my screen now. We're going to kind of switch into a live demo mode. And what I'm going to go through with you here is we're going to start here on production. And I want to first just call attention to the fact that in addition to managing all of this data, we have just textual information, tips, and that sort of thing that we want to share with folks. And so here in LabKey Server, we're able to add wiki type information. You can see I'm linking out to a quick start guide that we host within the site. We also have some support information that lives on board LabKey and is updated frequently. And we're also able here to provide reporting on site breakdowns by site and by sample type, and people are able to use a navigator to kind of show of the data sets we have, how complete are they, or how many data points do we have in each of those. I'm just quickly showing you all of this on prod to summarize that what we're up to right now is about 12 different data sets that I'll kind of walk you through. We have about over 4,700 subjects, and the samples are over 13,000 now coming from those subjects. And so all of that is here in our kind of prescribed solution in LabKey. And I'm going to switch gears into our QA environment because I've got all of our de-identified information here so I can show it to you in screen shares. LabKey itself lends itself to doing things like, say, copy production, put it on QA, and change all the sample IDs so I can show it to others, so that we actually use those tools to create this. But let me recenter here on the clinical data. And as you've probably heard in the news, if not other places, COVID manifests itself in a number of different ways. One being how patients come into the hospital, what phenotypes they're presented with. Often, if they have a really severe case, they're being given supplemental oxygen, and there are different levels of that. And of interest to our research consortium is that information alongside demographic information, lifestyle things like smoker status, and comorbidities like did you have hypertension or diabetes or other comorbidities that you were kind of reporting in with when you started developing the COVID-19 infection. So we have divided our data sets up among those different, capturing those different criteria here on the clinical side. And then also, while the patients are being seen in the hospital, they're also collecting clinical lab data. So we have different ways of presenting that lab data as data sets here in LabKey. And lastly, we're managing our samples. We're delivering some views that I'll show you. And in particular, I'm going to show this quickly just to kind of illustrate it. And again, this is a mock-up, but we mentioned that we have another source of data where our assay data is going. And if you can imagine 20 different labs all doing kind of their own thing in terms of creating their own lab assays on these samples, they're putting them in a system where they're curating them and that sort of thing and sharing them out among the consortium. That data, once it's curated, may come back into LabKey as its own data sets, but we're not there yet. And so what we're doing here is we're advertising at a patient and sample level whether there are different assays available, and then there's a link to that data. So this is kind of mocked up here, but I want to show you this is populated not by a person, but by an API with our other system. So we can have people come in here to a data set, kind of picture I'm in a regular user mode. They could click on a subject ID, and I've got one subject open here, and they see almost like a EMR all of the different data that has been collected for that patient. So I'm scanning through here, not getting yet into the details of what's in these data sets, but you can see of the ones that we've talked about, phenotypes and treatments and supplemental oxygen, I'm seeing all the data collected joined to this one patient ID. These are all the labs, so that's why it's scrolling so much. And then finally, I see the samples all collected. So this gives us a very powerful out-of-the-box method of saying, these are the different data that we're collecting. Users can go into a single patient level and kind of review everything. They can also see if assay data is available for that patient in this manner. Now, I'm going to switch gears here on you one more time and talk a little bit about as a typical LabKey user. You're going into these data sets and you have some features which I'm not going to espouse on very much, but you can create charts, you can do exports of particular interest to our group. That includes a lot of data analysts. You can export to a shell script that you can then take to something like R and make your own visualizations and connect it to other data sets. This is an important thing for us. Let me change hats on you one more time. And as we were defining all of these data sets, one of the things that we needed to do was define what goes in the data set. what is the structure, and provide some quality control on top of that. So here in our demographics data set, of importance to this particular COVID effort, we wanted to define each of these patients with whether their clinical definition was mild to severe. And you can see LabKey allowed us to put some descriptive information, so we have a shared understanding of what that clinical definition means. And also all the data that's used to kind of determine that clinical definition is here in the demographics data set alongside the typical things like age and sex. So some of the things that Adam mentioned in his updates talk was that you can use the UI not just to specify which fields are here, but important to us as we're looking at this architecture that I'm sharing with you is we could also apply reasoning to these fields to say this is a lookup value. You can only add certain values for the COVID status, or importantly, for the most aggressive measure of supplemental oxygen. So you can see a number of these are set up not only as text or decimal, but also as controlled vocabularies through this lookup function. And that allowed us to make a listing of these controlled vocabularies. I'm showing you a subsite where we do that in our implementation here. And I'm going to open oxygen delivery just to illustrate some of the things you can do that are quite advanced with this. So, you know, at the end of the day, we wanted to make sure that only certain values are enterable in our data set for this oxygen delivery field, and we define those here. But also, we want people to be able to analyze on that raw data or on a categorical definition of those. So if instead of getting all the way down to this granular level, we just want to say, I just want to see patients that have invasive mechanical ventilation or higher. This categorical metadata allows us to do that. So we're doing this on the list in LabKey, but if I go back to our data set, you can see that in my data set, I have the data that was entered, and then it's just simply showing me, since you entered room air, it is this category, or since you entered non-invasive, it is this category. And so this is shared out to our consortium, and they can do, this enables them to do more with the data. Similarly, I'm going to go to our phenotypes controlled vocabulary. This is something where we like using the human phenotype ontology, and this we're working very closely with LabKey to help define that ontology aware tool. This is just our sort of current approximation of that. It's not using a tool, it's just using a list like I just showed you previously. But here, our metadata, we're assigning a human phenotype ontology IDs, and we even make those linkable. So when we surface this in LabKey, our investigators can see the phenotypes that have been associated with the patient, but they can also be one click away from exploring those phenotypes. So this ability to define the data set, add definition information on a per column basis, and allow people to use that was an important part of our setup in LabKey. We use those same concepts here as we defined our samples. So we included different material types. Importantly, I want to show you that we, for many of our samples, can calculate based on a sample collection date, how many days was it from the symptom onset or diagnosis or admission to the hospital or ICU to that collection date. And this gives us sort of time points that are relative that can be shared among the many different sites that are contributing to this. And so this information is not really coming into us when the samples come to us, but we actually infer it from the other data sets I've shown. And we calculate it, and on a nightly basis, we actually update the samples. So as our users are perusing what samples are available, they can gain this information quite quickly. So I've shown you a few different contexts, data manager and typical user. And back in kind of that typical user role, they often have asked us for certain reports. And so our software development team has been able to deliver those. For example, I'm just showing you in summary that we are able to create a view of the sample data that I just showed you, which tracks everything at an aliquot level. But here we can roll all of those aliquots up into kind of an aggregate sample. So this is a custom view. It may not be applicable to everybody in the conference here, but this was important to us, and it was something just by using the query tools within LabKey and some of the LabKey SQL commands, we were able to build this the way our consortium wants to see it and deliver it to them through the same UI that they're using for everything else. Another similar thing, and I'll move on, is we're able to combine our data sets in a kind of pre-joined way with the samples. So here I'm showing another custom report where we take those data sets that you saw and we're joining them to sample information. And so if you can imagine, I can use out-of-the-box filter features here to build a cohort. Say I want to see only people over age 40 that required supplemental oxygen and had a positive outcome after that. I could apply those filters here and then say, so of those, how many do I have whole blood samples from? And basically build my cohort and then see what samples are available, all here in a single view within LabKey. I'm looking at the time, and so I think I'm going to move on here to our summary slides. Again, we've got a lot of content to cover here in these few minutes, and where we are today is, this was all stood up. None of us were what we'd consider LabKey experts back in May when this need was defined. But I would say in the months since then, our whole team has become up to speed. We've done 5 different publications out of the data that's in LabKey and being curated in LabKey, and others are on the way. We have enhancements planned, like integrating with REDCap, extending some of the dashboards I showed you, integrating with things like our server, and then of course just adding more data sets, including more genetic variant and biomarker data. Adam mentioned that there is this ongoing work with ontologies and the more robust data handling for data sets, that smart diffing that he referred to. Both of these are things that they've given us an ability to give them a voice, and we've expressed some of the things that we'd like to see as they develop this for everybody that's using LabKey, and we're quite grateful for that ability. And just bringing this to a close, some of our key takeaways for why this has been a success for our consortium. One, as I mentioned, this was stood up quickly, but allowed us to kind of progressively elaborate on the data sets. For example, as a new site came on board, we might need to add some more columns to capture the more diverse data that they're sharing with us, and we were able to progressively do that in LabKey. Another thing that contributed to the success was engaging with subject matter experts and our software developers, and really having a kind of all hands kind of approach among those roles to develop some of the features that we've seen. Prioritizing has been important because we couldn't do everything at once. Using APIs to do some of the automated work that I showed you has been important. And then finally, end user support, and even being able to deliver that support through wiki type content in LabKey was a contributor to our success. We'd like to acknowledge everyone in the Division of Intramural Research, from the stakeholders all the way to the data curators that are working on this huge effort, for engaging with us. Sandhya and I come from the Office of Cyber Infrastructure and Computational Biology, and we're thanking everyone from leadership that has helped with this to the data analysts and folks that helped with the implementation. And finally, the Advanced Biomedical Computational Science Group at National Cancer Institute that's working with that collaborative data sharing portal that I mentioned, and some of the tie-ins to LabKey. And finally, I think Sandhya mentioned it, and I've mentioned it also, the LabKey team, Adam, Hannah, Many others that we can't mention have just been really open to hearing our problems, helping us meet these challenges, and they've done a really great job for us over these past few months. So thank you, folks. I think that brings us to 1 o'clock, and we got a lot in within a few minutes.